All right, thank you everybody for attending and welcome to another talk in the IVEM. Today we're excited to host Yann Fichou. Yann Fichou is a professor at CNRS in Bordeaux, where he is doing research on amyloid and associated amyloid diseases, specifically the aggregation of tau fibrils, which he has taken a project which he has elaborately studied during his postdoctoral studies with Song Han, and he developed some very nice features, such as the idea of how to actually develop the background function for an amyloid. So as it elongates, how does elongation has an effect on the DIA signal or the EPR? And I think Jan will talk quite a lot about all those great features and developments he has been doing by himself now. And I'm looking forward to his talk. Thank you, Jan, for giving us a talk.
Thanks, Thomas. So yeah. Well, thank you for having me here. That's I'm glad to share some of the work we've been doing. So this is actually a bit embarrassing because I did present some 5 years ago in 2020, and it was already similar things. So I'm embarrassed because that should have been completely wrapped up. And well, now we have more data and understanding for sure, but still it should have been clearly finished by now. But well, that's how it is. So it will actually be the same theme than 5 years ago for those who were already there, but clearly with more depth in how we understand what we're measuring. All right. So, sorry. So, yeah. So it's titled how we can use DIR for amyloids. That's what I'm doing in general. Here, it's going to be targeted to something very specific that was just fun to walk and see really to what extent we can use these methods to characterize amyloid. So let me give you a bit of a background if I can change my slide. Yeah, so that's all the work is done on a protein called tau. So tau is actually an intrinsically disordered protein. So for those who are not in the protein world, it's you can see it as a biopolymer. Or a spaghetti if you're more in culinary art. So it's a spaghetti in solution. But it does have a function which is to regulate something called microtubules. They're fundamental for cells, and in particular in neurons. So it's actually long protein. You see, its native form is 441 amino acids, the longest. We actually use quite a few fragments, and in particular one of a shorter one, which is called tau 97. You see it here. Anyway, so tau is clearly the reason why tau is really well known. It's because it can form so-called amyloid filaments. Those amyloid filaments, they're found in neurons, and they're found in neurons that are dying in a disease where there is neurodegeneration. Those diseases, they're called tauopathies, and I'm sure you know Alzheimer's disease. It's one of them, but they're actually not limited to this. Quite a few, some 20 or so different tauopathies. So now, because cryo-EM came and it really provided a revolution in the sense that it could start to solve structure extracted from brain tissue. Then what was found with those studies that although it's the same protein, so it's always tau, it's always an amyloid, but the conformation that takes the protein inside within those amyloids is very disease specific. So it means first they're very different between diseases, and second there is no kind of overlap. They're very homogeneous within one disease, at least as far as cryo-EM can tell. So that's quite interesting because it brings you a link between our level of working at the molecular level of just structural biology all the way toward really the pathology, the pathological presentation of a given disease. So it's good for us molecularists because it gives kind of a biological justification why we care about those different structures. So exactly what do we care about? Well, we care about how this protein form amyloids. Sure, that's for sure. Then maybe a bit more subtle is how does tau form different amyloids? See what I mean? How it form those different structure? What really dictates the structural differentiation of tau? And I'm just showing here a few structure. They're perfect structure, but you know there's probably a gradient between all those different structures. So can we understand this with the bottom line that at the end of the day, in the tissue that we extract from human brain, we have those homogeneous structure here. So so that's the general question, and maybe giving to set the stage how we answer those questions. I need to give you a bit more information on those so-called amyloid aggregates. So they're highly ordered protein aggregates. So they're actually a stack of protein along the same direction. So that's why it's a filament. And then this stacking is in register and in a structure pattern that is called cross beta sheets. Okay, and the third point, it's supposed to be highly stable. If you draw an energy diagram, which I didn't put here, it's very low in free energy in principle. So that should be this arrangement should be super low in energy. So it should be highly stable and pretty much, well, often irreversible, at least for the pathological one. Okay, so that's just a background on amyloids. Then, so from there, I want to give you a bit. I want to talk about all stuff first, and that's really to give you an idea of where we're coming from and why you need to develop new methods. So this all stuff is something that was developed in the group of Songi some time ago when I was there. And it's the idea that because, well, so it's first the observation that was made that when we form those amyloids, you see them here. Let me maybe. Well, actually, I think you see my my mouse, so that should be fine. Sorry. So when we form those amyloids, we get a spectrum that you see here, and then we could decompose it into three components. Two of them are quite obvious. One is the monomer in solution. Another one is something much lower because we're forming big aggregates. But then there is also the third component that really come from the fact that we have in-register. And so this is theoretically expected. We have in-register packing, so this is very short distance. So we have dipolar broadening here that gives a very different line shape compared to regular nitroxide line. So from there, we could actually use the three components to deconvolve and to track those different components during aggregation, and so along aggregation kinetics that you see here, we could do this, and this is the actual results that we measure, where we can see these cross beta sheets based on the in-register packing. And to be sure that this is the case, if we moved away your label here, you move it away from the cross beta sheet pattern, then you lose this yellow contribution, which is the amyloids in register packed. Okay, so that's really the background here, and I would say at this point everything is normal, so to say. We expect this in register, it's all good. Now, what we found, again, back at this time, we found something quite puzzling, and so that's here. So what we found is that when we form those amyloid in vitro, and everything I've shown before is in vitro, and this is almost always done with a cofactor, it's heparin here. Okay, we always do this. Most of the people do this for the last 30 years now. And this was very often considered as just a catalyst, so it was no big deal in a sense to use it. And we will form those amyloid. I show you all the TM I show you before was with these methods. it diffracts perfectly at 4.7. So it's a canonical amyloid as far as we could tell. But so what we did here is we actually digest away the cofactor after that, and well, there is a story behind it, but the point was to get kind of the pure amyloid filament, just getting rid of whatever we added in the sketch. But what we found in that case is we completely depolymerized our fibers. We're losing our fibers, and when we remove this heparin. And so for me, that was kind of a kick in the dogma of amyloid being super stable, and pretty much you couldn't dissolve it. But so that was very surprising to me that when you remove those additional components, actually we're not stable. We're losing our amyloid filaments. So I think there is something here interesting, and so again, remember this because that's just a primer for what I want to tell afterward. So based on that, we wanted to investigate a bit more the identity of those filaments. So for that, deer is quite nice, and well, I actually don't have a proper introduction to deer, but what you if you don't know it, what you should know is that deer allow you to measure distance between spin label or between paramagnetic species in general. So if we place two labels on a monomer or on one molecule, then we can measure intramolecular distances, and that gives you information on the conformation of your protein. So that's really conventional way of using deer. So to do that, you really dilute because we don't want the spins we don't want the different monomers to interact too much with one another. So we dilute it with unlabeled protein so that we space away the labeled protein. And in addition, because we always anyway get some intermolecular interactions, we get rid of it in what we call the background in the data treatments. Background usually comes from intermolecular interaction. And so this, we don't want it. So if you do deer, you know it very well. If you know deer not so much, but you still do it, then this is removed even you don't know it. So this is always taken care of. So, okay, what we did from there, and again, sorry, this is old stuff, but it's a good primer. So what we did back there, we placed a few labels, and the idea was to have those different labels and to make those in vitro fibers and compare them with what you would expect from those brain extracted structure. Cryo-EM, obviously, we can compete with a full atomic structure, but we can map our distance on this. And what we found here is obviously the mapping is very different. So we don't form any of those two structures. That's what it means when we compare the black that we measure with the expected distance from the PDB. But here, the information I want to get to is the fact that at this time we conclude that because the width of our distribution was, we never got a narrow distribution as you would expect if you had a single defined structure. So we concluded that this is heterogeneous. They are structurally heterogeneous. And we actually, this is the same publication, we made this sketch. This is the table of contents sketch, where we draw heparin filament as something like this. Obviously, this is very hypothetical. We don't have direct data to show that. That was just a conclusion of the paper based on those distribution. But still, there were an idea of mispacking. And if you remember what I just said before, with also the fact that maybe we are not very stable amyloids, then this would also make sense. So there's really an interest to know whether what we're forming is how is it packed, and maybe there is mispacking that could lead to instability. So this is a big deal. All right. So with that background, which I wanted to put there because then there is a long way of some theoretical explanation which might lose you. But then remember that what we want to understand is some kind of heterogeneity in the in the amyloid. So that brings me back to my dear sketch. I already showed this slide. And what I've just showed before in my picture was the fact that every monomer might not be perfectly in register piled up. And so in a sense, this is a structural information that would appear not in this intramolecular dimension here, but along the packing, so along the longitudinal axis. And so as what do we get in along this longitudinal axis? Well, this is what we try to get rid of before this so-called background, or this so-called interaction between between molecules here. And so really what we wondered at this point is maybe instead of getting rid of it, we should try to understand it and see if from there we could get information of structural information along the fiber axis, and maybe answer some of our questions on how those different monomers are packed together. All right, so how do we do this? Well, so the background is here. So this is the expression typically we used. So I can give you a bit more information on what this means. So first of all, this is an expression that is derived from randomly distributed spins, considering that there is no specific interaction. So you have parameter D, and this is the dimensionality of your spin bath, whether it evolves on a line, on a surface, or in a volume, the solution, for example. And then you have alpha. And what alpha is really is the spin density, and in addition convolved with the pump efficiency. So you can have a very simple expression here. If you have D equal 3, that's the most common way, because this would be just a solution. And then it's slightly more complicated if you go to any fractal dimension D. But nothing that you cannot solve, especially because now we don't have to do the analytical resolution. We can just do numerical solving, so it's much easier, even though it's a complicated equation. Okay, so that's this those expressions are here. And at this point, I want to point out that there is something weird, at least for me, and maybe some experts will say it's obvious, but for me it wasn't obvious, is the fact that we have this idea of dimensionality here, but when you look at the theory behind here, the typical equation considers that we don't have orientation selection, so meaning there is no real information on how the spin are oriented with themselves, because we consider they have over orientation, and so it's power averaging. So for me, it was a bit weird to reconcile this with this notion of dimensionality, and so we've made some simulation I show to show that actually it doesn't originate from the fact that the spins are physically on a line or in a volume, but rather the fact that if they're on a line or if they're in a volume, just the distance distribution is different. Let me explain what I'm just saying. So those are simulation, purely theoretical here. So stay on the left panel with me. So what we do here is we place randomly distributed particle in a line or in a plane or on a volume, and then we compute only the first neighbor distance distribution. Okay, so we in those cases we only look at the first distance distribution. So there is no there is no geometry. Only we look at the first distance distribution when the spins are split like this. And you see that the distribution here is actually very different, whether in those three different configurations. So from that, we can directly simulate or calculate the dipolar evolution time that is the observable in deer, and we can fit these with this equation that I showed you before and recover the D that is fitted, and we found that we can completely find D equal 1, 2, and 3. So this long story is just to tell that this dimensionality really reflects different distance distribution, which originate from how the spins are organized, but there is no real orientation issues. Okay. So with that in mind, and that was really to be sure we understand all those systems. We played with it a bit, and well, I should say that when I say we was very much a work with Karen Say with finishing her PhD with Songi, and then we started together, and so it was very much a collaboration with her. So we played a bit with that. This is a standard 4-hydroxy tempo. So just a radical in solution. So you will expect this to be, just a bath of paramagnetic particles in solution. And so we did this at different concentration, and this behaved exactly as expected. We see D equal 3, and here until a certain concentration. That's because we have a steric hindrance. It's too populated, so then it's no longer randomly distributed. So we have a drop in D. And when you look at this alpha, you see that it's proportional to the concentration, which again is expected from this equation you see to the right. And the only thing we can conclude then, in addition, is that this pump efficiency here is not dependent on the concentration because it's linear here. And so we can actually use these methods, and that's what we did with Karen to calculate and to optimize your dear experiment, looking at the most efficient pump efficiency based on those measurements. But anyway, that's a different question, but it was interesting to based on this optimize the whole spectroscopic scheme. Okay, so this is for just tempo in solution. So now let's go back to our system of interest. It's an amyloid. So as I said, what we do here is we mix labeled protein with many more unlabeled protein. And so so we know those two are incorporated. That's what I showed you even with the CW. We know they're incorporated together. And when we change the ratio of labeled to unlabeled, what it means is we're changing the density of label in the fibers, right? You see, if we have 98% of unlabeled, then there is few spins along the fiber axis. If you have here 83%, then you have much more spins. So this is the data, then we fit it, and this is the result that we found. That was, I mean, this is the very first result I think we've got that really got us excited, because, well, first we get a reliable, very consistent dimensionality here, 1.5. And this I will come back to this after. And then we also get, as expected in a sense, but it wasn't given because it's a complicated system when we polymerize something. But we do get an alpha that is proportional to the spin label. So what this means is that here we can directly from there calculate the density of spins, and therefore we can calculate the spacing between each of those monomers in our assembly. So obviously this is here known already. It's an amyloid 4.7, but for any other system, that's an information that could be quite interesting, actually. So with that, we have actually played around and see what I want to show is the potential application of those kind of measurements. So for instance, we can move the label around. So these are 3 different label sites where we repeated the same experiment, and we measure each time the same the same Deer signal. And then we see that this dimensionality is self-consistent as long as you're within the amyloid core. And I should mention that this is something we know already, that this is within the amyloid core, and this is outside the amyloid core in the part that remain disordered. So you see that the dimensionality really jump up. So there is no doubt that those are significantly different. So we can probe effectively where is the amyloid core. Then we also try to do some kinetic experiment that is actually quite informative as well. So this is the kinetic of amyloid formation as followed by thioflavin T fluorescence that it stains for amyloid. Okay, so we can follow that's let's say a 10-ish hour process. And what we did here is we measure the same parameter, the same experiment before aggregation, and then we did it at 12 minutes after aggregation. So sorry, my line I have moved a bit, and the whole sketch is the whole graph is a bit weird, but 12 minutes is very short. So there is essentially no THT signal almost at 12 minutes. And then we did it later when, as far as THT is concerned, it's a mature filament. And so this is the results here, quite interesting. So this is before aggregation. So we see we're in solution, we can detect it very well. And then what was surprising is, well, first, then at the end, we see the 1.5, 1.6-ish that we used to measure. But then what was surprising is that already at 12 minutes, where we consider we don't necessarily have amyloids, we actually see already a big drop to 1.5 here, meaning that there seems to be very early oligomers that are not detected by fluorescence, but they're already partially aligned similarly to what we have in the fine fibers. Another quite important information we want to obtain, well, that is interesting to obtain on amyloid is the notion of protofilaments. So protofilament is essentially can be seen like this: is how many monomers do you have per layer? Is it just one protein that pile up onto each other, or is it several of them? So for instance, here, 2 of them in each layer, you have 2 monomers. And this is actually quite relevant, or it was proposed to be quite relevant as far as the pathology is concerned. So this is an interesting information, which is not always easy to obtain. So now, if you think about those 2 different configurations here, what you will find is that if you spin label, let's say 10% here, so and so you have 10% of your protein labeled here, so you have a given spin density, so number of spins per length of fibers. And if you do the same thing, but now you have 2 monomer per layer, what it means is that you're going to double the spin density along the fiber axis, along the fiber length here. So what means is per length of filament, you will have twice more spins in that case that you have in that case. So in principle, again, you might detect this in the alpha parameters I mentioned, that is direct measurements of spin density at the surface. So to test this, we've used 2. So tau-187 is what I've been using from the beginning. So that's the reference. But then we use a smaller fragment. And what we know with this smaller fragment is that this is supposed to be a 2 protofilament. So here you indeed are supposed to expect 2 monomer per layer, mostly because it's too small to fold back on itself. So we measure those 2, and this is very nice, because using the same spin dilution, you should expect similar alpha, but here clearly you see an alpha which is much bigger for tau 16, which means that really we're demonstrated here that this is a 2 protofilament, while this is only 1 protofilament. So we can detect really the quantity of monomer per layer that we have through this alpha parameter. All right. So this is now kind of the application we've played with and different information we think we can obtain by looking at this longitudinal access in this type of aggregates. Now I want to come back to this dimensionality that we consistently found to 1.5. And if it was a perfect line, we should expect 1, as I showed you before. So to understand why we got 1.5, we've actually used, again, we went back to a numerical simulation. And so we, again, this is numerical simulation. We simulate our line of spins by having different particles like this, and we would numerically make it paramagnetic. We will give each of those beads a probability to be paramagnetic, essentially, there with this probability P here. So what this is doing, essentially, at the end, it's just changing, well, from the experimental point of view, it's equivalent to change the ratio of labeled to unlabeled protein that we have. So how much spins per unit of length do we have? So that's the numerical simulation sketch here. So obviously we checked how it behaves, and here we're just changing the probability that we expect to be changing the density of spins. So we did the same. We extracted the first neighbor distribution here. And then we simulate the dipolar signal from there that we could that you see here, and from that you can fit it with our equation, so-called background equation, and you see exactly what you expect, meaning d equal 1 there, and you see the alpha that reacts directly to the density of spins that you imposed on your line. So that should be working. So with that, why do we said that? Because then we can test different hypotheses. And one major, not worries, but a hypothesis we had that maybe it's not randomly distributed. And in particular, if the spins interact with each other, and you could see 2 ways of interacting. One is repulsion, where because spins are, they're 200 Dalton, and you're asking them to be very close from one another, then maybe they will repulse them, meaning that statistically they wouldn't like to be, you wouldn't like to have 2 proteins labeled next to each other, and that's effectively a non-random event. So and the more repulsive side of it, if the label interact, if they like to be next to each other for some reason, then you will preferably have neighbor, very close neighbor along the fibers that are labeled. So we simulate this by having repulsion and attraction. What this means is that if you have a bead that is giving paramagnetic here, then you're imposing a penalty or different rules on the neighboring beads. If you want to do repulsion, then you're saying that you have a much lower probability to have a bead in the n+1, +2, n+3. Or if you want to simulate attraction, then you're increasing the probability of having a neighbor at n+1. So that's the results. Then we do all the thing we did before, which is to do the PFR, to simulate the dipolar signal, and to fit it with this equation and extract the D parameters. So when we simulate attraction, meaning that we impose here up to twice more probability to have a neighbor over the first neighbor, even second, even third. So third will be that if you have a bead at position n, then n+1, n+3, n+1, n+2, and n+3 have a much higher probability to be populated with a spin. So it's a very strong. So this blue here is a very strong constraint. and yet there is very little effect on it. So we don't think that this is very what could be happening. Similarly, with the repulsion, again, we say that there is lower probability to the extreme extent that we completely forbid any dimering spin. That's probability to 0, and we can forbid in the first neighbors, they wouldn't like to be next to each other, but even to higher neighbor. And you see that in this extreme case where 3 position will be completely forbidden, and then the dimension doesn't move very much. So we don't think that this non-randomness could explain our dimensionality. Okay, so then the next phenomena that we want to explore, and this will start to look back with my introduction. And this is what I said, there is a long way to go before I actually come back to my introduction on the importance of mispacking. So what we started to explore also is the fact that maybe our spin are not so well aligned with the amyloids. So maybe they have, they have some misalignment. And so this, again, from the simulation is fairly easy to do. We can, so what I did here is I allowed my bead to move on a disk like this randomly. So it's randomly moving on this disk, and I can increase the diameter of this disk or the radius to increase how much misaligned you are. And so this is what we do. You see the different amplitude of misalignment, and then we have the P of R. You see it actually changed quite drastically the shape of the P of R. And so this is dipolar signal, and this is the fitting results. And here you see that now you have a drastic change in the dimensionality, and actually you can start to explain the experimental observable that we had around 1.5, 1.6 by invoking a misalignment with an amplitude of 0 nanometers that you see here. So really, we think that maybe we're actually hanging to something that is probing imperfect alignments, which will be equivalent to, in my sense, to kind of a mispacking of these perfect amyloids. So that's our hypothesis. And to test this, what we did is we went back to the smaller fragments. I actually mentioned it a bit before, tau 16. And why is that interesting is because as you decrease the length of your protein, then you're very likely to decrease the chance of being mispacked, because you have less residues, so less conformational ensemble that configuration that you could have to make those amyloids. So clearly those shorter peptides, they're always much more ordered, just because you have less options in a sense to have alternative packing. So and you can see in the TN they're nice, and some maybe shorter peptides, but they've been even solved by crystallography as they're so well ordered. And so we did the same sketch here, and indeed we found that now we have a much lower dimensionality, closer to 1. They actually went even below 1, but closer to 1, let's say. So really, what I think is demonstrating here is that we can probe imperfect packing, and that those tau 187 triggered with heparin are indeed not really well packed. And the final proof of that is an experiment that we did recently, which is a seeding experiment. And so seeding experiment uses a template, so a small amount of a template in order to facilitate the growth of a protein around. So what we used a template of this tau 16 that we believe is well packed, and we hoped to see a templating of a better packing. So we seeded this tau 187 with something that is well packed, and we measured again these parameters. And what I'm showing here, this data is now the same protein tau 187 that you seeded with a better template, and we see although we don't reach the quality of the template, we do see an increase of dimensionality that we think really relate this idea of templating a good structure. All right, so that's about 40 minutes. So I think I'm way over time. So hopefully you've followed me so far. What I showed you is that this background actually might be carrying some information, and it's interesting because the methods that you have to actually look at the longitudinal axis are very limited. Often you just have to assume that they're fully packed, because otherwise you're just like I am, would you would just be blind by in the reconstruction if you're not. We can look at the spin density there, and with different structural properties, and the dimensionality we believe really can map the quality of the packing, which is not really good in the system I showed before, which is heparin-induced tau aggregates. All right. With that, I think I have a general thank you slide. Clearly this job was heavily started with Songi, and then we continue after that, and so that's something I'm excited to wrap up very soon. And I thank you for your attention.
Thank you, Yann, for the great talk. I really I enjoy that talk a lot because it really gives like a highlight look. There's so much more we can measure. We're not just restrained by one distance at a time because there's so many components. I mean, in this case, fine, we're looking at a deer, but there's so many components to it that we can harvest and describe biological functioning events such as amyloids, like how they're arranged and everything. So there's so much more to it. I really like that talk, actually. I appreciate it. Thank you.
Thank you. Thank you, Thomas.
Yeah. So with that being said, we come to my favorite part of the seminar series, and this is always a question and answer session. And while everybody is gathering their thoughts, I will just go ahead and plow right through. Quick question, Yann. With the offset, when you look at the paramagnetic offset from the center of the fibril, did you ever invoke that you have a turn? So meaning the fibril twist. Many fibers have a twist to it that would also cause an offset from the center because there's a twist around it unless the label is right in the center of the fiber axis.
Yeah.
So I figured, like, because you have that model already there, did you ever try to invoke a sine, cosine, whatever, like a twist function into it?
We actually haven't done it directly. It's only indirect. So this is it twisted. So the twist is it's actually quite long. I mean, the period is quite long. So the misalignment is actually quite small when you speak in terms of this scale here of a few monomers. And then, so this is really also captured in those simulation we've done on the misalignment, and you see the misalignment has to be fairly big. This is big. We are simulating very big disruption, several nanometers, up to 10 nanometers for every position. And so this is much heavier than what you would have when you have a slow twist. So I don't think this would explain this 1.5 for sure. Then, we should show it from simulation directly and to see if we could detect this. I think the more interesting question is maybe we could even detect a twist, and if we could find a relationship, we could even give an idea of what's the degree of twisting, which is very important even for like REM resolution. So but that's something exciting that I haven't tried.
Right, because I don't think there are many methods out there that can actually detect or quantify the twist. So that would be kind of weird.
If you're lucky, you see it under TM, which is easy, and that's that really depends if you have if your box in a sense is asymmetrical. Then if you have a rectangle, cross section, it's very easy to see, and then you can measure it under very classical TM. If it's if the cross section is square, then you're screwed because the PR is straight.
Right. All right, cool. Thank you, Yann. Anybody else? How about some other questions? Please just speak up or raise your hand, either one.
Yeah, I know this is very specific and unusual, unconventional way, so I don't know if it speaks to many people. Let's see.
Spoke to me, all right. Well, while we're waiting, how about I ask another question? Hey,
Sure.
so for the different length of tau, so one time you had the small tau and the large tau. And you said, I didn't really understand that. You said you used the small tau to make it more homogeneous than the large tau. I didn't really fully get that point.
Yeah, well, because the idea with this mispacking is that okay, some residues are not they're not perfectly packed in a cross beta sheet, but this can exist because let's say you have like a space. This can exist because you have many other residues to compensate. So some others around will be fixed and will give you the enthalpy that you need to really stay in place and packed up. As you decrease the quantity of residues, you have much less chance to rescue this mispacking. So mispacking cannot exist if you have a big gap where there is not these cross beta sheets anchoring, then the whole thing just cannot exist. So that's the idea. And so that's why all the crystallographies, I mean, this is from the 2000s, year 2010
Right.
maybe. They're all done on, I think, up to 8 to 10 residues because that gives you perfect packing. So that's the rationale behind. Then, well, you can even you can look at then cryo-EM and stuff like this. But clearly, those small peptides, they have to be more packed or they don't exist.
I see.
So yeah,
Cool.
that's the idea.
Then, well, one more question, just kind of curious, like actually in the beginning of your talk, and I'm kind of curious about your thoughts on that. So you were saying that the hapro in this case acted as a cofactor. When the cofactor was removed, the fiber was kind of falling apart in a way.
You're right.
What does that mean then in the grand scheme of things? That actually most fibers are rubbish because artificial, because we don't have the cofactors around?
Well, this is a long discussion. So you could argue maybe there is cofactors in the brain as well. They wouldn't be resolved. So you have a lot of densities in those cryo-EM maps that you have from the brains, and you have a lot of non-proteinaceous densities. So you could argue this way, or you could argue indeed that those heparin fibers, they're not relevant to biology or to the pathology, and clearly they're structurally very different. So for sure, they're not fully relevant. That's clearly the case. They're probably useful because they're still amyloids, so you still have some common pattern, but they're tau heparin amyloids, so you know they're this entity. They're not just a tau amyloids. They're tau heparin, and so whether this is interesting or not, that remains to be seen for sure.
Okay, I see.
Yeah.
Okay. Sorry.
Yeah. Well.
How about somebody else? Anybody else questions? Oh
Yeah,
wait, there's
I think
Veronica.
it's fine. I know this is
Hold on.
I know this is
Hold on.
very specific and
Veronica, why don't you go ahead?
Yeah, so thanks very much, Yann. It was a lot of stuff about background. And I guess what I'm wondering is, how does this really relate back to what physiologically is observed? And I mean, this is very specialized. I was thinking, is there a way to extend this to other systems, not amyloids? So, for example, polymer systems that adopt different, hexagonal packing and liquid crystals and things like that. Is this applicable to those types of systems as well? And then I have a question about the freezing, because if you have glycerol there, right, when you're freezing, how much does that impact these distances that you're observing and then what the background looks like, right?
Yeah. Okay. So there is a few questions there. So the well, I think this is so I guess the message I want to give is that these intermolecular distances that we very often want to get rid of might carry some information, and I think this is true also for any polymer, whether it's it's a covalent polymer or whether it's a polymer that comes from polymerization. Then yeah, absolutely. Then you can surely get information on the geometry of those polymers, or even on the surface. I mean, membrane surface that you can incorporate some spins, then you would be able to get some structural information, some arrangement on that. So I think this is very anything that have some self-assembly properties. Potentially this could be applicable. Then obviously this is not a very straightforward because it's a poor readout. I mean, it's only 2 parameters and it's a decay. So, it's very poor. It's even more poor than an oscillation, at least give you more information. This is just a decay, so we should be careful. But I think this study really demonstrates that we can still get information. So yeah, I guess that's my answer to part of your question. Maybe not all of it.
Yeah, that was sort of the application of it. What about the physiological relevance of the things you're studying, right? So you can't put spin labels in the ones that are assembling in vivo, right?
Yeah, I know.
Maybe you can, but I mean, what is the relationship, right?
Yeah. I mean, that's obviously we can't extract, but this packing is something very interesting, and why it's very interesting is because we really have issue to reproduce what we see in the brain in vitro, and we don't know why, in a sense. We don't know why, and so even when you do in vitro and you try to infect animals with that, you have much lower efficiency, and we don't know why. So gaining any information, and obviously now the very fashion readout we have is cryo-electron microscopy, and for good reason. But then you have absolutely no readout on this axis, how they're packed. You're completely blind to anything that wouldn't be reconstructed from a helical configuration. So I think this is an information that is unique to look at the quality, or one aspect of the quality of the fibers. And then we have all the seeding that I mentioned before, where we can demonstrate templating from brain extract into the material. Then this is just an additional readout on what's the final product we obtain.
And what about phosphorylation, right? So I thought that there are phosphorylation sites that are important in controlling the type of packing and things like that you get. And I don't think you've looked at that, right?
No, I haven't.
Okay. I'm not saying you should have. It's a lot of work to do. I get it.
Yeah. But you're right. I think those are all things that
right?
we can use these methods to look at.
Yeah.
For sure. But
Okay.
phosphorylation is
okay.
Thank you, Veronica. Thank you, Yann.
for the question.
Yeah, Muru, yes, please go ahead.
Yeah. Thanks, Yann. It's a great talk. So
Thanks.
my question is, heparin is an anticoagulant medication. It's an anticoagulant, okay? So, but in your case, it's like, in this case, amyloid aggregates. Heparin is a cofactor. So what that makes heparin is kind of different here, like, why? Here it makes like, it facilitates the aggregation.
Yeah. I mean, you're right. You're fully right. This is a very long discussion. So this heparin is coming from 1996 in a pioneer paper that showed that it can form amyloids. For 20 years. It was just assumed it was absolutely Alzheimer's fibrils. And now we realize that it's probably not Alzheimer's fibrils. There's been a lot of work because those sugars, they're always extracellular. And those aggregates of tau, they're intracellular. So there's been a lot of work to see whether when the protein is traveling, it might meet those heparin sulfate that are on the outside of the cells. And to some extent, some work has shown that there is this, even for internalization, you have this this interaction between the heparin sulfate and the protein. And then I think the most advanced explanation, which has absolutely no direct proof, is that you might undergo a conformational change because of this meeting with heparin sulfate, and then you keep this conformational change that is prone to aggregate, and it aggregate when it arrives in vitro in the cell. So this is very stretched. Maybe the most likely explanation is that it might not be heparin that trigger aggregation, but it might be something different cofactors, or even no cofactors at all. But heparin is just one of them, and you know, RNA would do the job as well. And RNA is already much more present inside the cell, although more in the nucleus, but still in the cytosol is there. So yeah, so I think heparin gives you a good principle, and it's very practical, very convenient in vitro. But it doesn't mean that it's the cofactor and it's directly relevant to what we see in the brain.
Thank you. Yeah.
Sure.
Great. Thank you very much.
All right.
And are there any other questions? Otherwise, I think we can let Yann go. Yeah, figure that's it. All right. Thank you everybody for attending, and I'm looking forward to seeing you guys in 2 weeks. Most importantly, thank you, Yann, for the great talk, and I hope to see you
My
soon
pleasure.
again.
Yeah,
All right.
absolutely. Thank you for organizing that.
No worries. Hey, Yann, did you get
Yeah.
an invite for Madhu?
Yeah, I did. So that's gonna be
Are you gonna did you tell him already that you're going, or what do you feel like?
yeah, I think
Yeah.
sure.
It'll be fun.
I'm very excited about it. So you too?
Yeah, he sent me an email like 2 days ago or something like that.
Yeah.
And then
Yeah, it should be quite exciting actually.
and then I go to India, right? Or anyways,
Yeah.
it'll be good. Like,
Yeah, this
let's
I know.
see.
I've never been there, and it's
No,
gonna be
I
interesting.
never have been either. It would be kind of fun to combine it together with like a family vacation away. Let's see.
Yeah, for sure. For sure. And
That works out.
I know Shital as well. He's very nice
Oh,
guy.
you do? I have no idea who that guy is.
Yeah,
Okay.
he was postdoc with me. We were postdocing for 4 years together, so I know
Okay.
him very well. He was with
Okay, so
Songi.
that's really good.
Yeah. So I'm pretty excited about seeing him also.
Okay. So that's
He's
good.
not doing biology at all. He's doing very much more material science and NMR also.
Okay. Have you talked to Sonia lately? I haven't talked to her for a long time.
She was actually in Bordeaux last Friday. I invited her for. I had a defense for. We have to defend some diploma, some habilitation.
Okay.
So she was part of my jury on Friday.
Oh,
So
really? That's kind of nice to
yeah.
have a friend as a jury.
Yeah. We have we had the whole thing. It's kind of HDR. It's like a PhD defense. You have to prepare a manuscript and you defend in front of a
Oh, really?
6-person jury, and then they write a report and they say, oh yeah,
So did you pass?
Yeah,
Oh,
sure.
of course. So you're a full professor then, or?
It's a formality at the end, but the pain is
Yeah.
to organize everybody, have everybody
how-
coming, you know. But
how
yeah,
is
but
she
it was
doing?
fun. So she was there.
Oh, how is she doing, Northwestern? I heard like last time I talked to her, which is God knows how long ago, she was still having a hard time getting everything set up and all this stuff.
Yeah, no, I think she was she was doing well actually. She's she's Tongi, so she's all over the place doing many things at the same time.
Yeah.
But no, I think she has settled now, pretty much.
That's good. All good. There's
So
all right.
Yeah.
Well,
Hopefully, we can meet all of us soon.
yeah, it will be kind of fun. It's kind of nice of Madhu to set up all this stuff. It's gonna be pretty nice too.
Yeah.
Sorry, man.
absolutely.
I'm a little bit like all the allergies are kicking my butt. It sounds like you're a
Well,
little
I
bit
have to go
sick as
anyway. I
well.
have a meeting now, so but
All right.
thank you for the invitation, and
No
let's discuss soon.
All right, sounds good. See you, buddy.
All right, goodbye.
Bye.